Sir, we are live now.
All right. Very good morning, everyone. Welcome to all the MOOC students and participants who are joining us today for the live session number 1. So we have Interactomics course as well as the Bioengineering course. The students have already started weekly assignment and the videos to watch. What we thought for today's live session that we are going to give you an overview of something new which is not covered in the class or your course, but will be related to what you are learning, and should add your knowledge in such a way that how some of these fundamental concepts which we talk, how they come from the laboratory-based work, right? So today we are going to take you from our lab, which is the clinical proteomics lab, and going to show you different technologies which can be utilized for biomarker discovery and other applications. So today's topic and the theme what we decided was essentially how to identify and discover the novel biomarkers and potential candidates using various type of proteomic technologies. I'm Sanjeeva Srivastava, your instructor for this MOOC course. Along with me, Dr. Arya Banerjee, who is the main TA for this course, is also joining, and some other student and TAs from my team will be also giving you some demonstrations. So coming to the biomarker theme, this is something which is very important whenever we go for any kind of disease, right? To be monitored or we are unsure about what is going wrong. So the clinicians, the doctors, the path labs, what they test for, they look for some sort of biomolecule, right? So they look for what is the abnormality happening in our body based on some marker. And those biomarker, which we say the biomarker, are essentially going to reflect us what is the a disease condition look like, or what kind of infection we may have, or other kind of metabolic disorders if one is going through. So biomarker could be defined as a biomolecule which is detected in our body fluids or tissue and could be associated with the special processes, could be in the monitoring the normal responses or looking at the abnormal disease conditions. So from the, if we are suspected patient, we are going to look at some of those biomarkers to determine that, is that really required to have more advanced testing for any type of specific disease. Think about, for example, the elderly men who may be suspected for the prostate cancer. They go for a test known as the PSA, prostate specific antigen test, which is essentially looking at the antibodies against the PSA protein and trying to measure those and see whether there is elevation of this particular marker, PSA biomarker. Similarly, for many of the other cancer type like breast cancer, ovarian cancer, etc., there are set of markers from the EBNA protein to the CA protein to looking at different type of P53 and other panel of the protein as a biomarker, which are being utilized to do the early screening. So biomarker is utilized for the detection of the diseases, but also could be utilized during the patient treatment. For example, if a patient is given a set of the chemotherapy drugs and whether they are responding well or not, how to monitor that response, whether the patient who has been given a treatment has fully recovered or may have potential to show the disease recurrence. Again, so the markers could really guide us at different stages, right? So based on that, one could think about biomarkers for different activities, like the diagnostic biomarkers, which can be utilized for detection of the disease early, prognostic biomarker, which is going to tell us a disease is a very mild stage at the early level or at the very advanced stage or very severe form. For example, in the COVID case, everybody who had the COVID was cautious to know that whether they have the mild form of COVID or the severe form, right? Like as I said, like whether disease could recur as well in case of cancer, how disease is going to progress, whether somebody will survive. Many of these things are actually being monitored using some biomolecule, especially the protein, or even also the metabolite and lipid and other kind of biomolecules. Predictive biomarker, again utilized to looking at the patient assessment that the treatment has gone very well. Now these type of proteins are showing that the levels are very much in control. But if they are going to show the shoot up the level, it may be indicated that it is going to come back or going to recur. So at a different level of clinical decisions, these biomarkers becomes very, very important. and therefore discovering them, it becomes quite useful for taking the decisions, whether it's a diagnostic, prognostic, or therapeutic decisions. So biomarker study design, it starts from defining the clinical problem. What is the actual clinical disease or the stage which we are interested to investigate? We have to then think about how to design that study, because we need to have sufficient power analysis using statistical predictions to, let's say, have maybe 100 controls and 100 disease samples. What could be a good control for a given disease is very challenging always. For instance, if somebody you are going to investigate a biomarker which you are going to screen against the malaria patients, you are looking for biomarker for malaria, the best control may not be healthy individual. Best control will be other febrile infection, people who have other type of fever, but may not have the malaria infection. They may have dengue, they may have leptospirosis, chikungunya, they may have jaundice, some other type of infections, which is causing similar symptom and giving the fever response, febrile response, which is like acute phase responses will be activated over there. So that will be the right control. For a cancer, for example, somebody is having a brain tumor, and you are going to compare them with the normal individual, that may not be the best control again, right? So looking at an individual which is you're collecting samples from the brain, but different disorder, or you're looking at other type of cancer. For example, for a brain tumor, you are looking at meningioma, like glioma as a brain tumor, you're looking at meningioma as a control, because meningioma is another type of brain tumor, but origin is different. Or one could also look at other type of cancer, for example, oral cancer or some other type of cancer. So again, what you are looking at, cancer itself will have many hallmark genes and proteins going to be expressed. If we could nullify that, reduce those, so okay, what kind of a specific biomarker we can identify, which is very unique to the type of brain tumor or type of disease which we are looking at, right? So looking at selection of the patient, study design, the matched controls, the type of controls, different type of positive, negative control, all that becomes very relevant and important for designing our study. Then next comes to which type of technology we want to use, whether we want to utilize the two-dimensional gels, we want to utilize the mass spectrometry, we want to utilize microarrays, we want to utilize other analytical technologies. And what should be our biological material? Should be a plasma sample, serum sample, tissue sample, saliva sample, cerebrospinal fluid. What kind of sample we should be looking at? Because again, looking at the sample which is most proximal to that disease. For example, for a brain tumor, looking at cerebrospinal fluid and the brain tissue of the tumor will be most relevant. Saliva or the plasma may not be most relevant sample, right? So you have to think about that kind of study design, and then look at the statistical study design. What should be the best way of looking at different cohorts and looking at the power of the analysis? So I hope this gives you a bird's eye view that when we say biomarker, it is not so straightforward just to go and identify something very quickly with one shot. So you need to look at multiple consideration for study design, to sample, to patient, to looking at your analytical approaches. What today we are going to focus on is going to look at mainly, while we'll give you the bird's eye view of different technologies, but we are also going to give you a little more zoom in focus on mass spectrometry based proteomics workflow. And let's say if we are looking at the tissue sample, then the workflow will be slightly different. If you're looking at the serum or plasma sample, you have to remove some abundant protein, that will be slightly different. But then finally it comes to the instrumentation part, the LC-MS, which we are going to show you different type of mass spec in the lab, which will be quite similar. We have different Orbitrap and timsTOF and quadruple instrument, which we are going to demonstrate you, which is resulting into different type of data analysis to look at the data dependent acquisition or data independent acquisition based analysis. We will focus more onto the latest approaches of DIA based approaches. And again, then making sense of the data requires a lot of time. We will give you some, kind of preamble to that, but more detail we will cover in the next live session. So I'll stop right now here because I just wanted to make sure that we are having sufficient time for our live session from the lab. So I request Dr. Arghya now to take over and show some of the sample prep and the, different technologies in our lab. And then we will talk a little bit more toward the end about applications of this. So Arghya, over to you.
share my slide. स्क्रीन आ रहा है वो बताओ.
Make it full screen, Arge. I can see your slide.
Okay, very good. So let us go with the whole overview of the protocol. So this is the protocol in the clinical samples. The samples can be of cell or tissue origin, or a blood sample or a blood sample. The basic step, the first step is of isolating the proteins out of it. Okay, so in the cell or the tissue, what we do is generally prepare a cell or the tissue lysate. Once the tissue lysate is being prepared, Once the tissue lysate is being prepared, then the proteins are quantified. Once the proteins are quantified, we go for the simple step of digestion. And once the digestion has been done and peptides are isolated, we generally go for the HRMS acquisition, followed by the data analysis and visualization. Generally, for the protein extraction, these are the reagents used. We can use the cell pellets or the tissue. A lysis buffer is required. That lysis buffer can be an urea lysis buffer or the Proteomics kit own buffer. We add a protease inhibitor cocktail to inactivate the serum protease, which are basically in the tissues, followed by the bad food quantification. So here we will move on. Here we will move on with the video how the things are done.
Arge, you're not audible. Arge, please turn on the audio and then show demonstration.
Yeah, so this is the Eppendorf tube in which the 50 microgram of the tissue samples have been acquired, and in this 50 microgram of tissue samples, we have generally added the protein cocktail inhibitor. In the role of the protein cocktail inhibitor is to destabilize or inactivate the protein serinase, the protease serinase, which results into degradation. Because you have to be more cautious when you do a phosphatase study. So when you do a phosphatase study, we have to be more careful so that the proteins or the tissue should not get degraded. So now let's come here. What you can find. Once the tissue samples has been collected, what we can do a qualification check. In the qualification check, like you can do a SDS page. You can do an SDS page followed by, yes, you can find like this is the apparatus where we cast the gel, Once the SDS page has been casted, you run your samples into the apparatus, the running apparatus. And once the sample has been run, you can also run the gel. So this is how the gels look like. So you can clearly find these are the gel bands, what you can see. And these are the band patterns of the clinical samples, what you have find. So once this is being done, once your gel is clear, you can proceed with the digestion, or else you can also perform two-dimensional gel electrophoresis. So in the SDS page, the molecules are separated on the basis of molecular weight, but with the help of the two-dimensional gel electrophoresis, you can see these are the IPG strips what are placed here. You can first separate the proteins on the basis of their isoelectric point. And once the proteins are separated on the basis of their isoelectric point, you can then load the gel into an SDS page where the separation happens according to their molecular weight. So maybe in the next session, next video session, we can discuss more detail about the SDS page. So this is how you can find once the tissue sample is being measured, we generally lyse the tissue samples for the cell lysate. So this is an tissue bit box, Proteomics tissue bit box, which uses magnetic bits for the lysis. So you just open the tissue bit box. So this is how the tissue bit box apparatus look like. There is a magnetic field on bottom. You can place your samples in the tissue bit box. You can place your samples in the tissue bit box containing the magnetic bits, and you can place it inside. And yes, you can place this thing inside. You can choose the time and the temperature depending upon the tissue. We have generally selected the 10 minute lysis. Like you can select the time and temperature on the basis. So we have generally selected for 8 minutes. The normal soft tissues can be well lysed for 8 minutes. You can set the standard as low and high based on what kind of shearing pattern, magnetic shearing pattern you want. So I can set it for low and then just use starting samples. So you can see the time technology that the time has initiated. The time has initiated and within 8 minutes you can done. So I will just stop this reaction for now. So once your tissue is being lysed, once your tissue is being lysed, once your tissue is lysed, now the main thing is for the digestion. We will show how the proteomics digestion happens. So I will request Khushi, my TA. बताओ पहले थोड़ा सा introduce करो.
My name is Khushi Tiwari, and we are for digestion. We are using Proteomics-based digestion. So here is our tissue sample, and in this we take 100 microgram of tissue sample, and we add 50 microliter of lysate water and incubate 95 degrees Celsius for 10 minutes. And here we put 95 degrees Celsius.
So we just incubate the samples at 95 degrees Celsius. So basically the lysis, the first step consists of the reduction and the alkylation. The lysis buffer itself has the solvents which are equivalent to the TCEP as well as the alkylation like indole acetamide, which does the reduction as well as the alkylation. The main logic of here is like after 10 minutes we take out the samples. The main logic here is like we prevent any sort of like the reduction happens due to the TCEP. Once the reduction is done, we prevent the proteins from again refolding it back by doing the alkylation. Once the reduction and the alkylation is being done, now it makes a way for the enzyme which is trypsin to enter into the proteins and then perform the digestion. So here we are now adding the trypsin solution. So once the trypsin is being added, again it is incubated for 4 hours. We'll just take the sample. We will heat incubate it in the hot air bath for 4 hours. So during this process, the trypsin incubates. During this process, the trypsin goes and cleaves the proteins which are present in the unfolded state, which results into the formation of the peptides. Once the peptide is being taken, like after 4 hours of digestion, once the peptide is being taken out, so these are the peptide samples. We will add this top solution. The logic of adding this top solution is quenching the reaction. What do you mean by the quenching? Is like the overall trypsin activity can lead into degradation or the complete degradation of the peptides. To stop that trypsin activity, we generally add a lysis or we can totally dry the samples. So this is how we have added the quenching solution. We will incubate it for some time. And once the incubation is done, we'll do the peptide quantification. So this is an plate reader, technical plate reader, what you are seeing. इसको खोलो. So this is how the plates look like. So this is a micro drop. So this is a micro drop plate reader. We generally place our peptides here. So we can place the peptides here. Once the peptides are being loaded, when the 2 microliter of peptide is more than enough to be loaded, we place the plate, we place the micro drop plate and let it move inside. So basically this is our in-house generated protocol. So this is how the parameter settings looks like. So what is the basic logic? Just take out the plate layout. So this is how the plate layout looks at. The basic logic is that we measure the absorbance or the intensities at 280. We measure the peptide abundance and the intensities at the 280 measure it at 280 as well as the peptide at 205. Once the intensities are being measured, we dry these peptide samples totally. Once the peptide samples are totally dried, okay, we reconstitute in the LC load or the LC loading buffer. We can, so one step we missed is like we can quantify the protein samples initially with the Bradford. So this is how the plate layout generally looks like. You can quantify your protein samples with the Bradford with the standard PSA samples. And this is on the basis of the coloring or the intensities, the things are changed. Okay. So you can easily quantify. Another method which can be used for this kind of quantification is, I will just show it, is the protein microarray. What you can find, like this is a plate reader. This is a protein microarray. You can load your samples or the tissue lysates into these protein microarray chips, which consist of around 20 to 30,000 proteins, which are spotted in duplicates. Like this is how a chip looks like. So these are the UProt chips. So proteins are imprinted. And once the proteins are incubated, you can easily scan it in the scanner. So this is the protein microarray scanner. And this is how the scanner looks like.
So coming back, I will just flash through this slide. So we have seen how the cell pellet is being taken for digestion, followed by the magnetic bead beater, tissue bead box. Once the tissue bead box is being done, we quantify the protein samples. Once the protein samples are being quantified, we check the solubility with the SDS page. We check the quality with the help of the SDS bands, and then we see a protein concentration estimation either with the Bradford or the BCA. You can see like this is how a calibration curve looks like. With an R square value of 0.99, you find the unknown samples, the concentration of the unknown samples, and this is how the process of the digestion takes place. So once the lysate is being quantified, you know the protein quantification. You can take 50 or 20 microgram of the proteins or peptide samples. You can add TCEP, you can incubate it at 37 degree, which what happens is like we have used a Proteomics kit, so we have used a temperature of 90 degree, but generally TCEP helps in the reduction. Once the reduction is done, we perform alkylation. Once the alkylation has been done, it is ready for the enzyme to be treated. Now, once the enzyme is being added, the trypsinization happens. So this is the Proteomics protocol, what we have generally followed. We have taken 100 mg of lyse proteins. We have incubated for 10 minutes. We have again resuspended into the digestion buffer, which consists of the trypsin. Once the trypsin has been used, we have used 100 microliter of stop solution to check the reaction of the trypsinization, followed by the wash 1 and wash 2. So we parallelly go for peptide quantification. What we have done for the peptides, and next is like once the tips are prepared, we will go for the mass spec quantification. So professor can give the brief initial exposition of the mass spec. What are the kinds of mass specs which are available for this kind of analysis? Back to you, professor.
All right, thank you, Arya. So now since you have seen at least the broad workflow that how starting from any sample, especially the tissue lysate or the cell lysate, you can reach till the peptide level and quantify the peptides. So from that, then you have multiple options in which way you can acquire the data from mass spec. One could utilize different type of LCMS based platform. It depends on what is the goal, right? If goal is to look at unbiased analysis of identifying the new biomarker or new target, then you'd like to look at as comprehensive coverage as possible. If the goal was to look at only specific targets or validate the target, then you'd like to do the very specific focused target analysis. So let's first think about, you are looking at the unbiased global analysis to look for the new candidate. So here aim will be, can we get like, thousands of protein and as many protein from my given sample as possible? Because more the sample I get in, then more my probability of getting the good candidate will be there. So this sample we can run in different type of mass spec data acquisition mode using different type of mass spec. I think shortly Agya will show you a couple of Orbitrap instrument which we have and which is going to give you idea that in which way LC-MS could be utilized to show the broad workflow and broadly the data acquisition part. But idea is that we are going to utilize a workflow known as the DIA, data independent acquisition, which is going to systematically look at the entire M by Z window of my peptides, whether from the 300 to the 13 or 1400 mass to charge ratio, and acquire systematically data from all the isolation windows from the low precursors, low intensity versus the high intensity, but everything from all the windows. So that usually gives us much broader coverage and improves the issues what we usually face with the missing value data, which comes from the label-free quantification type of analysis. So a typical Orbitrap based instrument, which we are going to show you, Orbitrap Fusion, over there we try to inject 1 microgram peptide. So you have already quantified the peptide. Now we are going to show you how to inject the peptide and then going to utilize the nanoliquid chromatography followed by the MS for data acquisition. Typically, we use 2 hours gradient to improve the coverage and therefore utilizing all the skill, we are then trying to get 3,500 to 5,000 proteins using this technology and workflow. Recently, we have been more fortunate to have additional instrumentation, which is the timsTOF mass spec, which is also capable of doing 4D omics. This instrument in a much lower amount of peptide, which is 200 to 300 nanogram peptide, with around 44 minutes of gradient, we are able to utilize that to get almost 8,500 to 10,000 proteins. So much broader coverage, but that's not something which we're going to show today. That will be the part of the later live sessions. Today's live session will be essentially showing you some Orbitrap instrumentation right now. And so I request now Argya to go and show some injection of the peptide on the Orbitrap Fusion instrument which we have. This is one of the tribrid mass spec, which is capable of doing a different type of application because we have the Orbitrap and quadrupole and ion trap, which is part of the mass analyzers of this instrument, which is tribrid mass spec. So over to you, Argya, to show the live demonstrations of Orbitrap Fusion, followed by the Q executive for different applications.
So this is how an Orbitrap fusion looks like, and this is the nano component. like which nano LC has been injected. So the nano LCs have the solvent. First is any nano LC, first of any nano LC, you will have two kinds of solvent, which is solvent A and solvent B. The solvent A may be of organic phase, which consists of 80% of the ACN, followed by solvent B, which is 0.1% of FA. So this is how a nano LC component. So this is how a nano LC component looks like. So there is a demo version of the nano LC, one more square nano LC, where I am going to show you. Like we can inject the tray out. So here is how the tray looks like. So these are the solvents, these are the trays. You can inject the samples inside the tube. So this is how a real peptide, after your peptides have been quantified, okay, you can just inject the samples. You can just inject the samples of the peptide here. So at a stretch, you can easily go for 96 well samples. So once it is done, you can check on all the routine maintenance, like you can do the calibration, you can prepare, you can test, you can service the nano LC, but that is not required for as of today. We'll go to the home and we can start, like we can start the general procedure, that if you can check like if the plate or the buffer is ready or not. Okay, so once this is being done, the analysis starts. So what the nanoANSI does is that it segregates the proteins, it segregates the peptides on the basis of two components. One is the pre-column and one is the separation column or the trap column. Basically, you have to be more cautious that the no samples, no fault traces are being available. Otherwise, it will choke the column. The columns are of micron size and the solvents which are flowing through it are purely clean, devoid of any fault contamination. The pre-column, the pre-column generally is a preventive column which prevents any debris or any solvent which to come in. So you can find like this is the pre-column. This is basically the ion source of the orbitrap. So this is a nano LC EV spray ion source. So you can see the pre-column. So this part is the pre-column. You can see the pre-column. First, the samples are eluted. Once the samples are eluted, it reaches to the pre-column. Then this part is our main loading column or the mass spec column. So once it passes through the pre-column, it enters into the mass spec column, and you can find that there is an isothermal capillary tube or the ITC tube inside. The samples which are injected, it is bombarded inside. Once it's bombarded inside, it goes to the orbiter or the mass analyzer. The mass analyzer analyzes the proteins, and you can see a chromatogram. You can see a chromatogram like that. So this is how a live sample run acquisition happens. The top is the MS acquisition or the chromatogram, and the bottom is the MS fragmentation. With each MS or the MS ions, we can open a chromatogram and show. So this is how a chromatogram looks like. So this is a 120 minutes gradient. So we can zoom in a region, a small region, and we can show. So for each MS, we are about to see. So this is how it looks like. You can click on any peak. For each MS, we can easily see 10 to 20 MS acquisition. That means the data has been very goodly occupied. Okay, the histogram has been very goodly accepted. So this is how the samples or the data is being generated. Once the data is being generated, we can analyze it with the MS Fragger or the easily routinely available software, free software such as DIAN. So this data will now reveal like what all proteins or the which have been isolated through global discovery, as Professor said. One more instrument which is very robust is Q exactive. Is the Q exactive? You can see our mass spectrum, our mass spectrometry handler which is being opened. So you can see. So this is an UHPLC. So this is an ultra high liquid performance chromatography. This is the sample loading tray. You can find it has several colors such as blue, red, and green. At a stage, 200 samples can be easily floated in a day. Okay? So I will just close the sample compartment. generally 4-degree temperature is being maintained. So once you put a command, the sample compartment and the samples are injected inside. The solvents you have like if you have a quaternary pump, you can have many solvents as an A, B, C, and D. Generally, A is an acetonitrile or methanol, like an organic solvent, and B is in water. The solvent flows inside. The injection, the solvent flows inside. The injection picks the column. The injection picks the samples, which is placed inside the tray. Okay, inside the tray, and then it injects into the mask. So this, what we have earlier shown is the nano LC source, but this is an hazy source, which is connected to the HPLC. Once the hazy source, the samples is getting injected, you can see the cone inside. You can see the cone inside. The sample travels through it. You can easily, so yeah, so you can easily see the cone inside. The samples get injected through this cone, and the sample acquisition happens in the mass spec. Can we open any chromatogram? Any chromatogram? So yeah, so this is how we can do metabolomics acquisition with the help of the Q exactive, which is also an ion and mass spec. So once you have identified like what all proteins are there and what all metabolites are there in your sample, we can go to another mass spec which is specialized for triple quadrupole or the TSQ altis. TSQ altis. So here you can see like this instrument is the TSQ altis, and this instrument is an Vanquish. Like this is an advanced version of the HPLC. In the Vanquish, you can easily see like these are the sample compartment which is maintained at 4 degree, and you can place the samples inside. You can place the samples inside. So here you can see there are samples with the tube. You can place around 200 samples per day. Once the sample is being injected, once the samples is being injected, this is how a quaternary pump looks like, and it travels through the HPLC column. So you can easily find this as the column compartment. Once the samples is getting injected into the HPLC column, it again comes to the hazy source, and from the hazy source, it goes like you can see there is an so this is how a hazy source of the compartment looks like, and from the hazy source we inject the samples and it goes into the market. So this is how a chromatogram. So this is how the chromatogram of the acquisition looks like. So these are the each transitions. So you can see in our routine quality check, this is the Resapine data. Generally, these are the Resapine quality control checks which have been done. See, so these are the transitions. So we will study in great detail in your tutorial. Like these are the transitions, and these are how the pics have been taken. Okay, so each transition with the pic cursor and the master ion circle. So apart from this, like this is a very excellent quality for the validation, like the validation. Once this is being carried out, the data is really good for SRM or PRM related validation. Apart from that, there is one more instrument which is the BLI or the biolayer interferometry. Here, what happens, you can find the easily the when you are dealing with the antibodies and the antigens, you can easily find the avidity or the binding efficiency of the antigens and the antibodies. The samples are embedded into the golden chips, and you can, based on the piezoelectric potential, you can easily find the affinity and avidity. So just to recall what we have seen the proteins can be easily monitored at a global level on an untargeted discovery with the help of DIA method, data independent method, which we can discuss in detail in other classes with the help of the orbitrap fusion. This is an orbitrap instrument which is placed coupled with the nano LC, and easily it can go for around the 120 minutes gradient. You can get a very good chromatogram. Once you are done with this, you can also validate your metabolites with the help of another orbitrap instrument, which is orbitrap Q exactive. coupled with HPLC with a short minute gradient of 10 to 12 minutes. And finally, once you know your target, which targets or which proteins can be of potential, you can then analyze with the help of triple quadrupole, which is analogous to Western or Southern blotting. You can validate your individual proteins in the peaks. You can find the biomarker panel, what pseudo biomarker panels, what can be the exact biomarker of. So this is the demonstration of the three instruments. So this is your post EA. So back to you, Prof.
All right, Arya, thank you. So I think all the student and participant, you got some flavor of how these approaches can be utilized to look for the new targets, and these are very unbiased approach. So therefore, if you are looking at something, a problem for which there is no existing biomarker available, then you need to first start with a global approach, as you've just seen. And from there onward, then you can try to select the panels and then go for targeted approach, which is we showed you on the triple quadrupole instrument. You can also do validation using the Orbitrap technology, which can be done with the PRM approach known as parallel reaction monitoring. What we showed you was the MRM approach, multiple reaction monitoring. Let me share you my slides again and just give you some idea that, okay, what all we are talking in terms of the different type of data acquisition, different type of data analysis, how that actually helps to understand some biological problems and clinical problems. So for example, this is the one work I'm showing you. We were looking at the biomarkers for vivax malaria. So almost 200 plus patient samples we analyzed from different clinical hospitals. And idea was to look at the vivax related protein, parasite related proteins, which are secreted in the human blood. So it means the parasite protein should be such a concentration that is we are able to detect them from the blood itself. So it means these proteins will be in very high concentration, secreted from the parasite and being reflected and identified from the plasma sample. If we can do that, then these will be good candidate towards going for the different type of RDT kits. So with this intention, one of my PhD student at that time, Apurva Venkatesh, she looked at these patient plasma sample. We did both parasite proteome and the host proteome analysis using LC-MS based approaches. And many candidate biomarkers were identified, but then idea was how unique they are only for the vivax malaria and not seen even in the other malaria type, including falciparum malaria. So then we did a lot of sequence analysis and bioinformatic analysis, and that helped us to reduce the those proteins to these 5 candidates, which we further validated using the targeted approaches which we just showed you. So now you see, like, you start from the unbiased large number of clinical samples, look at the potential targets, do bioinformatics, see what is novelty, validate them, and then take for the next level of translations. Additionally, we were also interested to know that what type of complications malaria patients have and whether we can identify some sort of specific pathways to depict that biology. For example, many individuals who go from the non-severe falciparum malaria infection to the severe form of the falciparum malaria, some of them goes the complication in the brain known as cerebral malaria. Some show anemic response, severe anemia, and some show other type of complications. So looking at different type of pathway, which was a very comprehensive work done and was published in one of the Nature Communication Biology paper, where we were able to show first time, like, the integrin cell surface related proteins and those markers were more enriched in the cerebral malaria, while looking at some of the hemostasis related pathways that are more relevant for the severe anemia and many other platelet factors and the various type of acute phase response proteins in other type of severe complications. So again, just looking at this broad picture, we were able to get the different mechanistic insight of different type of complications. Additionally, we also compared the dengue and the vivax malaria and different type of pathways involved in that. To save time, I'm not talking about all of the biology, but it gives you idea that how these type of discovery work takes it to the next level of understanding the clinical problems or the biological problems. During the COVID time, again, the whole world was unknown for the COVID. We had no idea. So we were looking at COVID negative individuals and COVID positive individuals. Within COVID positive, we had the mild and the severe type of complicated individuals. We looked at various type of biological sample from the nasopharyngeal swab sample to the plasma sample. I'm showing you the plasma sample workflow here. Idea was, can we find out some biomarker which could be indicative of how a patient is progressing from the mild form to the severe form of the complication. To do that, then we did the same type of LCMS-based workflow, acquired a lot of data, analyzed the data, did the target validation, and then look at potential drug candidates. After doing your data analysis, which Arya will talk to you shortly, what you will realize that you still have a lot of candidates, which are potential candidates. And those candidates, we can then do some machine learning to look at how to narrow down that panel, which panel is the most interesting and influential panel for our analysis. For example, apolipo-B and fibrinogen gamma chain came as a very important proteins, uh, along with two other markers which we identified and made a patent for the prognostic test for looking at how disease get mild to the severe form. We were also in turn able to identify the disease pathways, which is making the COVID-19 very complicated and severe form. Many type of platelet degranulation pathways, some of the leukocyte activation pathways, complement pathways, they were quite perturbed in as the patient is moving from the mild form to the severe form of disease. So I'll just, for the time being, stop here and let Arya talk to you about the data analysis pipeline. Data analysis is much more detailed, eventually when we'll talk to the next session, but at least he will give you the primer of it and some bird's eye view of how data analysis steps to be performed. And then at the end, I will talk to you about some of the ways in which way biomarkers are reaching to the FDA approvals and looking at clinical translation activity. Arya, please go ahead.
So here you can find, like, once you have so here you can find, this is the data pre-analysis pathway. Like, once the data has been generated from the frac with the help of mass spec, you can usually analyze it with the frac pipe. The frac pipe is an open software, you can usually download it. So this is an frac pipe interface, how it looks like in the workflow, as we can do both the DDA and the DIA. So in the later classes we will show you like how the DIA data looks like what is the difference between DDA and the DIA. So DIA gives more coverage. We have selected DIA spectral library for quantification. Your system requirements, it should have minimum of 10 GB of RAM with parallel, that can do a parallel processing of 8 files. And here finally, uh, you can add your files. Once you add your files, like the raw files, .raw files, which are being generated from the mass spec, you can sort it by the file by name. Like if you have a control 1 2 3, here only two files I have uploaded, like the HILA and the QC. You can do uploading on the basis of the names, like file by name of control 1, control 2, control 3, and disease 1, disease 2, disease 3. As most of the individuals, like most of the clinical samples which we deal with, the human samples is so we have selected the human FASTA. But based on your clinical study, you can also select the the mouse FASTA, or else the yeast FASTA if you are expressing or coding or any cloning experiments are being done. So everything can be applicable. And if you don't have the database, you can make your database and upload it. Along with the reviewed FASTA or the reviewed sequence, you have to add the decoys and the contaminants, which will help us to identify which proteins are the contaminant proteins. So this is the database setting. Once the database setting, we will go to the MS Fragger session. We will run the MS Fragger. Here the precursor mass tolerance has been taken to -20 to +20 ppm, which is on the stringent side. You can also take 0.1 to -10 to +10 ppm, but generally in DIA we go for -20 to -20 ppm. In the enzyme section, you have to select the trypsin. Like this is the trypsin, strict trypsin, because we have used only trypsin. You can select the other options also, such as lysi and any, if you are doing any combination enzyme for the digestion. So here, as we have selected only trypsin, it will be used only trypsin. So it is strict trypsin. followed by the cuts at the end of the lysine and the arginine region. So as we from the basic chemistry we know, trypsin cleaves the lysine and arginine end. The number of miscleavages you have kept it, we have kept it as 1, but you can, in the strict condition, you can do it as 0 also. But we generally keep 1 to 2 miscleavages. And methionine modification and urea modification we have kept. The peptide length we are considering only if it is more than 7 amino acid, like 7 to 50 amino acid. Once this is being done, the peptide mass range can be 500, and based on that, the validation will be done. So we come to the quant DIA method. We select diane. Okay, so what it does, that this fact pipe has a free search algorithm which is DIANN diane algorithm, which is inside it only. We select the search algorithm or the diane. For the FDR, we select 0.01, and we generate the MS stat output, which is also encoded, like also saved in this. Once this is being done, the output data is being shared, like the output, you save the output folder, and once this is being done, you just run the file. So now you get a P cross N matrix which consists of the protein list as well as the intensities with it. You have to perform a secondary data analysis. So what does secondary data analysis means? Your samples may consist of like the missing values. Then your samples can be run on different days. That is due to variability can come. You have to normalize. Either that can be due to the experimental handling or due to time. Once the normalization is being done, you have to do a data integrity check or data integrity check. So these all steps we can do. So meta-analytics, like in some of the workshop, like in some of the live session, we will give you a strong demo. We can do this multivariate analysis. It is a freely available software. You just click on to the statistical analysis on the meta-analyst. You select the peak intensities. When the peak intensities is being selected, you move on to the data integrity check. So once the data integrity check is being done, you do a remove the missing values. In any biological studies, we take samples or we carry forward the data only of the samples which consist of at least 70% of the occurrence. If you are having missing 30% value, it can be imputed on the basis of the 70% of the population. But as a good biology, but as a good rule here, like in our lab, we generally remove the samples which are not occurring, like which are occurring not occurring in less than 70%. So 70 to 100% is our acceptance ratio. We remove the features which are less than 30%. We can perform various kinds of low repeatability filters such as standard deviation or interquantile. Each has their own modeling. And finally we do the missing value imputation. So you can find, like, this is how a difference between the missing percentage and an average value percentage used. Like, once your missing value imputation is being completed, we move on to the multivariate analysis. The multivariate analysis starts with the normalization. Normalization is generally to reduce the experimental or the variations which has been caused due to reproducibility, which is occurring in every 6 months. Like if you are planning a stability experiment where your clinical samples or the biomarkers are run in 1 month, third month, fourth month, and so on for the repeated number of points, okay, or any experimental deviation. So you can easily select whatever median, whatever normalization we look like. Or in our proteomics cases, median normalization mostly satisfies the criteria. You can find, like, these are the normalized. Before normalized, you can find two hump, and after normalization you can find a single hump which has the perfect central tendency aligned to it. Log transformation, generally the mass spec data which are generated are into the 6 digit. Our eye can be difficult in managing those data. So what we do, we generally change into a log transformation to the base 2 or base 10, so that the number comes in the 2 digit. Data scaling, you can perform auto scaling or you can perform auto scaling. Once that is being done, this is how a graph looks like. So this is the normalized data. Once the normalized data is being done, we move on to the statistical data analysis, where you can first predict the PCA, which is PCA plot, which is an means unbiased, like the unbiased or ungrouped analysis, where you select the best eigen values. Like you have a features, basically features are mentioned, and based on the eigen values of these features, your best classifiers are identified. So I just submit my samples in simple the PCA plot. I just complete principal component analysis plot. I just submit my samples, and based on the eigen values, the best classifiers are selected, and then your samples are segregated whether it is coming good or not. So here you can see like this is of one pool, and this is for second pool, and this is for the third pool. It performs an individual cohort. The PC we generally run for the 5 PC values. Generally PC1 and PC2 gives the most of the satisfied data, which consists of more than 50%. If your PCA plot is not coming good, you can go for clustered, like the supervised clustering, where you mention like this is my control and this is my disease, or group 2. Now on the basis of these two groups, now identify which are my best eigenvalues. So your clustered PCA like the PCLA or PCLDA can identify the best classifiers and give the best classifiers out of it. After that, we perform a correlation plot. Like you can find, this is a correlation plot of the cell line, which is of sensitive and resistant, and you can find a perfect correlation happening at the diagonal, which where r square equals to 1. In biology, anything which comes about zero point 0.5, in biology, if the r square is more than zero point 0.5, then it is positively correlated, and if it is equal to 1, it is strongly correlated, and if it is minus, if it is 0, then it is not correlated. In biology, we generally take a value of r square more than zero point 0.5, like r value of more
Yeah.
than 0.5 to 1.
Yeah, sorry for interrupting, but I think, you have covered at least the broad overview. We have to stop it here. I'll just quickly wrap up with the pipeline currently with the approved biomarkers. So, just for all the participant, I think, you will be curious after doing all this hard work, where do we stand on actually, some of these markers reaching to the clinical testing, right? So what has been more recent success that panel of biomarker, for example, if you look at OA1, this includes 5 proteins, CA125, prealbumin, apolipoprotein, beta-2 microglobulin, and transferrin. Based on this, it is already FDA approved that one could decide that there is a sufficient ovarian cancer risk in women with the adnexal mass. Which type of markers can be utilized, like in a Roma, for example, test, can we look at whether there is a high risk of ovarian cancer in the pelvic mass? Which individual should be done the surgery, or which should be just kept on the watch and wait? All that can be done using these type of biomarker. Looking at hepatocellular carcinoma, liver cancer, markers like DCP and AFPL3, they are indicative. Prostate cancer, I already told you earlier. Ovarian is another test which is deciding the prediction of ovarian cancer high risk in the women. So what you see, this knowledge what we have generated. I have already shared the slides. I don't know if my slides are not shared with everybody. I'm just kind of.
Yes, sir, it's visible.
It is visible, right? Okay. So, yeah, so just to conclude here, so what you see, a lot of biomarkers have already reached to the clinical translational ways. And the pipeline for looking at biomarkers there are different phases, like, phase 1, phase 2, phase 3, phase 4, phase 5, which goes through various type of approval stages. And based on those approvals, then after reaching to the phase 5, only then they reached out to the actual biomarker for the clinical testing to be done. And over as one of those biomarkers. So again, a student and participant, or maybe some of the student of the medical background who might be keen to learn more about it, they should visit this website of EDRN, Early Detection Research Network in US, which is from the NIH. That will give you several of the ongoing tests and the approved tests. For example, there's a test, many of the clinical laboratory improvement amendments are being updated. Some tests being done, like a Vidisha test for the breast cancer based on the 19 protein biomarker panel. And there are many other, success story, which I think I don't have time, but you should follow it up and see many biomarkers are reaching to the clinical translation, and get more detail and overview of that from the NIH NCI website directly. So I'll just stop it here. And I hope our idea was today to give you the workflow from starting from the sample to the getting the different type of instrument platforms, in which way your consideration should be for good clinical study design, looking at different type of parameters, different analytical platform, the samples, the controls, and what type of instrument to be chosen. Many of these things are, which comes as a part of the experience which you are acquiring in the courses which you are taking. And once you have acquired the data, then you can do extensive data analysis. Then you need to do more independent validation, blinded validation, take more sample from different cohorts. And if it passes all of that, then independent testing to be done before the regulatory approvals are being obtained. So this kind of pipeline for biomarker, which is complicated but really essential, lot of money and investment and the intellectual work is involved in doing this activity. And many labs worldwide are investigating these, and many companies investigating the biomarkers to look for different problems from diagnostics, prognostic, or therapeutics. I hope you have got some insight and some new knowledge from today's live session. We'll be taking more of the queries in the forum, and we also have a Telegram group to take more of your queries and your any doubts you may have. And we'll be looking forward for the next live session, which we have in the next month, where we are going to cover you some more exciting technologies and more exciting workflows. Till then, we're going to stop you here. And Argya and the TS, thank you for showing the technology